Your heart is racing. You think it's because you're in love. It isn't. It's because your brain filed this person under emergency. I want to be precise about that because the precision matters. What you're experiencing, the accelerated heartbeat, the heightened alertness, the inability to think clearly about anything else, is not a romantic response in any special sense. It's a stress response. The same neurological and physiological machinery that fires when you're in danger fires when you see someone you find compelling. Your heart cannot distinguish between a threat and a person. It receives the same signal either way. That's not a reductive claim. It's the beginning of something more interesting. Because the question of why the brain routes romantic attraction through the threat response system, why love and danger share the same machinery, is one of the most revealing questions in neuroscience. And the answer leads somewhere that neither biology nor physics has fully resolved. Let's follow the signal. Start with the heart itself, because most people have a confused picture of what it does and who controls it. The heart has its own electrical system. The sinoatrial node, a small cluster of cells in the right atrium, generates electrical impulses that trigger each heartbeat. This system is intrinsic. It doesn't require the brain. If you cut every nerve connecting the heart to the central nervous system, the heart continues beating at roughly 100 beats per minute, which is its intrinsic rate without any external input. Your resting heart rate is lower than that, 60 to 80 beats per minute for most people. The difference is the vagus nerve. The vagus nerve is the main conduit of the parasympathetic nervous system to the heart. At rest, it applies continuous braking, what physiologists call vagal tone. It holds the heart below its intrinsic rate. A resting heart rate of 70 is not the heart running freely. It's the heart being continuously restrained. When the sympathetic nervous system activates, two things happen simultaneously. Sympathetic signals reach the sinoatrial node and accelerate it. And the vagus nerve withdraws. The braking is released. The heart accelerates from both directions at once. Acceleration applied and braking removed at the same moment. What triggers sympathetic activation? The brain, specifically regions that assess significance, that decide whether something in the environment matters enough to require the body's full resources. And when you see someone you're attracted to, those regions activate. The heart doesn't know why. It doesn't receive context. It receives a signal, accelerate, and it does. You feel the acceleration. You interpret it as love. But the interpretation happens after the fact. The body moved first, before you'd consciously finished forming a thought about the person in front of you. The sequence precisely is this. You perceive a person. Visual and social information enters through the senses, is processed in layers, first in subcortical regions, then in cortical ones. Somewhere in that processing, before you've consciously formed a complete perception, a structure deep in your temporal lobe has already made a judgment. The amygdala, a small almond-shaped structure, bilateral, one in each hemisphere, that functions as the brain's rapid significance detector. It processes emotionally relevant stimuli faster than conscious awareness. Studies using masking techniques, presenting a stimulus for milliseconds, too brief for conscious recognition, show that the amygdala responds to emotionally significant faces even when the subject is unaware of having seen them. Before you know you find someone attractive, the amygdala has already flagged them. The amygdala activates the hypothalamus. The hypothalamus is the brain's command center for autonomic function. It coordinates the body's response to significant events by signaling the autonomic nervous system. It sends signals down the sympathetic chain. The sympathetic chain reaches the adrenal glands, which sit atop each kidney. The adrenal medulla releases epinephrine, adrenaline, into the bloodstream. Within seconds, epinephrine is circulating throughout the body. It binds to beta-adrenergic receptors on the sinoatrial node. Heart rate increases. Force of contraction increases. Cardiac output rises. You feel your heart. Simultaneously, norepinephrine floods the prefrontal cortex, sharpening attention and focus toward the significant stimulus, the person in front of you. Cortisol begins to rise. Dopamine is released in the nucleus accumbens. Activity decreases in brain regions associated with critical evaluation. The capacity for rational skepticism about the object of attraction is biochemically reduced. All of this in seconds, most of it before consciousness has caught up. What you experience as falling, the sudden quality of it, the sense of something having happened to you rather than something you chose, is accurate. Something did happen to you. A cascade of neurological events that began before you were aware of them, that produced a physical state your conscious mind then interpreted. The feeling came last. Here's what I find most revealing about the mechanism. Epinephrine doesn't carry a label. When it binds to the receptors on your heart, the heart has no way of knowing why it was released, whether the amygdala flagged a threat or a potential mate. The downstream signal is the same molecule. The receptor is the same. The cardiac response is identical. This isn't a quirk. It's a fundamental feature of how the arousal system works. In 1974, psychologists Donald Dutton and Arthur Aaron conducted an experiment at 2 bridges in British Columbia. One bridge was a narrow suspension bridge, high, wobbly, with low handrails over a significant drop, physiologically alarming. The other was a solid, low, stable structure. An attractive research assistant approached men on each bridge and asked them to complete a questionnaire, then gave them her phone number in case they wanted to discuss the study further. Men on the frightening bridge were significantly more likely to call her afterward. They were also more likely to include sexual content in a projective story task they'd been given. The physiological arousal from the frightening bridge, the accelerated heart, the heightened alertness, had been attributed in part to the woman. The racing heart was the same whether caused by the bridge or by attraction. The brain attached different stories. This is called misattribution of arousal, and it reveals something important. The emotional label we apply to a physical state is not delivered with the state. It's constructed afterward by a brain searching for the most plausible explanation of what the body is experiencing. You feel arousal. You ask implicitly, what caused this? The most salient thing in the environment becomes the answer. The bridge study worked because the bridge made men aroused before they met the interviewer, and the brain resolved the ambiguity by attributing the arousal to her. This means you can manufacture, to some extent, the physiology of attraction by engineering the right context. Physiologically arousing situations, novelty, mild threat, physical activity, can be misattributed to a companion. The brain updates its assessment of the companion based partly on the arousal state the companion was present during. Why did the brain build the systems this way? Why use the same machinery for danger and for love? Because from the perspective of an organism concerned with survival, both represent situations of high significance with uncertain outcomes. A predator might kill you. A potential mate might change your reproductive future entirely. Both require heightened alertness, rapid response capacity, focused attention, the suspension of normal cognitive operations. The same physiological preparation serves both. Building one system for significance and applying it to everything that's significant is more efficient than building separate systems for every type of significant event. The brain didn't build special romantic hardware, it repurposed its threat hardware. You are in love the way you are in danger, through the same machinery, generating the same signals, producing the same physical state. But epinephrine and cortisol only explain arousal. They don't explain the specific texture of romantic love, the wanting, the preoccupation, the sense that this particular person is irreplaceable. For that, you need the rest of the chemistry. Dopamine. The nucleus accumbens, the brain's primary reward structure, activates in response to the sight of someone you're attracted to, with an intensity that researchers initially found surprising. The activation profile resembles responses to addictive substances. Not metaphorically, the same reward circuitry firing with similar patterns. Dopamine in this context doesn't produce satisfaction, it produces wanting. The craving, the intrusive thoughts, the preoccupation, these are dopaminergic. The reward system is responding to the anticipation of a reward it hasn't fully received yet, which is the state that produces the most intense dopaminergic activation. Early romantic love is neurologically closer to the anticipatory state of addiction than to the satisfied state of any completed reward. This is why early love is consuming. The dopamine system is running at high intensity, focused on something it wants and doesn't fully have. The brain keeps returning to the stimulus the way it returns to anything that activates the reward system at that intensity. Then oxytocin, released during physical touch, during sustained eye contact, during the kinds of social interaction that signal safety and bonding. Oxytocin has a specific relationship with the stress response. It buffers cortisol, reducing the threat quality of the arousal. When two people who are attracted to each other make physical contact, some of the epinephrine and cortisol arousal shifts in character. The threat signal decreases, something warmer takes its place. The racing heart is still there, but it's starting to feel like something other than danger. Touch is the mechanism that begins converting the threat response into attachment. Without physical contact, the acute stress response of attraction persists at a higher intensity. Touch changes the chemistry. Vasopressin works differently from oxytocin. It's associated specifically with long-term pair bonding. In prairie voles, one of the few genuinely monogamous mammals, blocking vasopressin receptors eliminates pair bonding behavior. The animals still mate, they don't form exclusive partnerships. In humans, variants in the vasopressin receptor gene correlate with relationship quality and commitment behavior. The molecule that encodes this specific partner maintained over time is vasopressin, and serotonin, which behaves counterintuitively in early love. Serotonin levels drop. The neurological profile of early romantic love includes reduced serotonin that resembles the profile of obsessive-compulsive disorder. The intrusive, repetitive, returning thoughts, the inability to stop thinking about a particular person, is neurochemically similar to obsession in its clinical sense. Not pathological, but using the same machinery. The combination produces the specific experience. Dopamine drives the wanting. Oxytocin promotes the sense of safety and bonding. Vasopressin encodes the specificity. This person, not just any person. Serotonin reduction creates the focus that feels like preoccupation. All of it running on a sympathetic nervous system that the amygdala activated before any of this chemistry had a name. The acute response doesn't last. Most people in long-term relationships know this. The racing heart, the obsessive focus, the consuming quality of early love diminishes, usually within 1 to 3 years. What's happening neurologically is that the brain has updated its model. The person was once novel, uncertain, unpredicted, requiring the high arousal significance response the amygdala fires for the unknown. After sustained exposure, they are known. The brain has built a detailed predictive model of them. Prediction violations, the moments when someone does something unexpected but retrospectively right, become less frequent as the model becomes more accurate. The amygdala quiets. The acute epinephrine release decreases. The dopaminergic craving becomes less intense. The heart rate normalizes. This is often described culturally as the love fading. The neuroscience suggests a different interpretation. The acute stress response is decreasing because the brain has completed the work it does for novel, significant stimuli. The oxytocin and vasopressin architecture of stable attachment, which produces a quieter, more consistent experience, becomes dominant. The relationship hasn't weakened, its neurological profile has changed. There's a consistent finding in relationship research that couples who engage in novel, arousing activities together report higher relationship satisfaction than those whose shared activities have become routine. The mechanism is misattribution of arousal, running in the same direction as the bridge study. Novelty and mild physiological arousal generated by the activity gets partially attributed to the partner. The brain, searching for what's significant in this moment, updates its assessment of the person it's with. You can, in limited ways, reintroduce the chemistry. Not fully, the novelty of encountering someone for the first time cannot be recreated. But the arousal system is still there, still attributable. The brain still constructs explanations for the physical states it finds itself in. Every measurable component of romantic love has a neurochemical correlate: amygdala activation, epinephrine, cortisol, dopamine, oxytocin, vasopressin, serotonin. We can identify when each releases, in what sequence, in what regions. We can map the full cascade from first perception to stable attachment. And yet the description feels incomplete. Not wrong, incomplete. Knowing that dopamine drives the wanting doesn't explain what it's like to miss a specific person. The dopamine description applies to every person who has ever experienced early romantic love. It doesn't differentiate. But the experience is entirely differentiated. It's not romantic love in the abstract, it's this person whose absence has a specific weight, whose presence changes something in the quality of ordinary experience. The neurochemistry explains the mechanism. It doesn't explain why this particular configuration of neurons responding to this particular stimulus produces something that feels irreplaceable rather than interchangeable. This is the hard problem of consciousness, applied specifically to love. The explanatory gap between the physical description and the subjective experience doesn't close when you add more biochemistry. You can add every molecule, every receptor, every activation pattern. The question of what it's like to love someone remains outside the description. I spent my career believing that a complete mathematical description of a system was equivalent to understanding it. The Dirac equation completely describes the electron, its mass, its charge, its spin, its behavior in fields, its interaction with other particles. It predicted antimatter. It is precise to a degree that continues to be confirmed experimentally. Does that mean we understand what an electron is? I'm less certain now than I once was. There is a difference between a description that predicts behavior completely and an explanation that accounts for the thing itself. The equation is the first. Whether it's the second is a harder question. The complete neurochemical description of love predicts behavior with reasonable accuracy. It explains the racing heart, the preoccupation, the shift from acute arousal to stable attachment. It describes every measurable component of the experience. But the person in front of you, the specific, particular, irreplaceable fact of them, That is not in any equation. The racing heart is the easiest part to explain. What the heart is racing for, that remains the hardest problem there is. Not because the science is wrong, because the science, however complete, is a description, and what you're experiencing is something that happens to the one experiencing it, from the inside, in a way that no description from the outside fully reaches. The heart races, the chemistry cascades, the brain constructs an experience of significance and attachment and irreplaceability from signals that don't inherently carry those meanings. And somewhere in that construction, in the gap between the signal and the meaning, is everything that matters about it.